Today we will be beginning Chapter 6 and we're going to discuss neurotransmitter systems. There are 5 key types of neurotransmitter receptors I'm going to discuss today, including acetylcholine, norepinephrine, GABA, glycine, and glutamate receptors. I'm also making a handout with all of these different types of receptors, and I'll post that to Canvas, so keep your eye out for that. So we've already talked about agonists and antagonists, but I just want to refresh your memory. These are drugs that can come and function either like neurotransmitters or they can block the receptor and inhibit effects. So agonists are drugs that can come and bind to a receptor and mimic the naturally occurring neurotransmitter, which activates the receptor. However, an antagonist is a drug that will come and bind to the receptor and simply block it. This will produce no effect. Today we're going to talk about different types of agonists versus antagonists. So let's look at 2 different types of agonists. First, I'm going to discuss nicotine, which is a nicotinic acetylcholinergic receptor agonist and induces excitation in the postsynaptic neuron, and muscarine, which is a muscarinic acetylcholinergic receptor agonist and induces inhibition in the postsynaptic neuron. Nicotine is often taken in through tobacco use. We know that nicotine is an agonist at the nicotinic acetylcholinergic receptor. Now, I know that sounds like a mouthful, but in my previous lectures, I've talked about how acetylcholine can sometimes inhibit the actions of the postsynaptic neuron or excite the postsynaptic neuron. Well, this is a case where nicotine is an agonist that's actually mimicking acetylcholine and inducing excitation in the postsynaptic neuron. Throughout my lecture, you're going to see these symbols, either plus signs or minus signs, and they're going to denote whether that particular agonist or antagonist is excitatory or inhibitory. Nicotine works at the neuromuscular junction. This is where all of our neurons synapse onto muscles. There are also nicotinic acetylcholinergic receptors in the central nervous system, so that includes the brain and brainstem, and these are involved in the addictive effects of tobacco use. So let's go through this step by step. We know that nicotine binds to and activates ligand-gated acetylcholinergic receptors in the skeletal muscle. This receptor is ionotropic, meaning that nicotine binds to the ligand-gated ion channel and the channel opens. This allows extracellular sodium to come inside of the cell. When this extracellular sodium comes into the cytoplasm, it's going to induce depolarization and thus generate an excitatory postsynaptic potential. When you look at my handout sheet, I have examples of all of the receptors I'm going to be talking about today in today's lecture. And so you can see all of these drawn out and follow along through the handout. Here's the second type of acetylcholinergic receptor I'm going to talk about today, and that is the muscarinic acetylcholinergic receptor. Now, this is a G protein coupled receptor that utilizes the shortcut pathway. Don't worry, I'm going to explain that in a minute. But first, I want to introduce muscarine, which is an agonist that binds to the muscarinic acetylcholinergic receptor and mimics the naturally occurring neurotransmitter at play, which is acetylcholine. So muscarinic agonists activate this receptor and help aid in digestion, heart rate regulation, and memory formation. I'm specifically going to use an example of how it functions at the heart muscle specifically. So this is a G protein coupled receptor that is inhibitory, meaning it's going to stop activity in the postsynaptic neuron. So when we're talking about the muscarinic acetylcholinergic receptor, remember that these G proteins are in the heart muscle, and they're activated when acetylcholine binds to this muscarinic receptor. Similarly, the agonist muscarine can bind to this receptor and mimic the naturally occurring neurotransmitter, which is acetylcholine. So here's the image that I drew out. This is also in the handout. Muscarine comes and binds to the G protein coupled receptor, which can induce a conformational change. We know that G proteins are made of multiple subunits, including the alpha, beta, and gamma subunits. We also know that these subunits are anchored to the phospholipid membrane with these lipid anchors. Hopefully this should be review for you guys. Essentially, after muscarine binds to the G protein coupled receptor, it's going to influence neighboring ion channels. For example, this potassium-gated ion channel nearby is going to open up its gates. And when it opens up its gates, it's going to allow intracellular potassium to leave the cell. Because potassium is positively charged, this is going to induce an inhibitory postsynaptic potential in our postsynaptic neuron due to this negative membrane potential. So thus, this potassium is increasing inhibition. Think of the parasympathetic response in Otto Louis' frog heart experiment. As I have the frog here as a reminder, you can remember that in his experiment when he discovered the neurotransmitter acetylcholine, which he called Wegestoff, he realized that acetylcholine was the chemical that was responsible for reducing the heart rate after the vagus nerve was stimulated. If you remember in this experiment, he stimulated the vagus nerve of one frog heart that was connected to a second frog heart, and liquid or PBS solution from the first frog heart could move to the second frog heart freely. So thus, once the first frog heart generated acetylcholine following vagus nerve stimulation, it also reduced the heart rate of the second frog heart, even though it didn't receive stimulation on its own. And this is all due to action at the muscarinic acetylcholinergic receptor. So remember Otto Louis' frog experiment when you're studying this G protein coupled receptor. Now let's look at two different types of antagonists. First, we're going to talk about cobra venom, which is an acetylcholine receptor antagonist, and another acetylcholine receptor antagonist known as curare. Both of these prevent acetylcholine from doing its job at the neuromuscular junction. Cobra venom is an antagonist at the nicotinic acetylcholinergic receptor. Now, we've already talked about this receptor in the context of nicotine. Nicotine is an agonist, meaning it's mimicking the naturally occurring neurotransmitter acetylcholine at this receptor. In contrast, cobra venom is an antagonist, meaning it blocks this receptor and prevents it from doing its job. The bite of the Taiwanese cobra also results in the blockade of neuromuscular transmission, but yet through another mechanism. So one of the active compounds in the Taiwanese cobra's venom is called alpha bungarotoxin, and this is a peptide that binds so tightly to the postsynaptic receptors that it takes days to be removed. There's usually no time for the removal though, because this toxin prevents the activation of the nicotinic acetylcholine receptors, and this paralyzes the respiratory muscles of its victims. So thus, these nicotinic receptors play a vital role in our respiratory health, and the bite of this Taiwanese cobra can be fatal. Another example of an acetylcholine antagonist is curare. So curare is a poison, and it's traditionally used in South America. Natives in South America will actually add curare to their arrow tips to paralyze their prey. If you're familiar with the show The Green Arrow, you know that Oliver Queen has experienced curare many times as he's stranded on an island for 5 years. Curare can be very deadly. It binds tightly to the acetylcholine receptors on skeletal muscle cells and blocks the actions of acetylcholine. It therefore prevents muscle contraction. Now we're going to discuss toxins that affect presynaptic neurotransmitter release. Because these toxins only affect the presynaptic cell, they are not labeled as agonist or antagonists. First, we're going to discuss Botox, and then we're going to talk about black widow spider venom. So botulinum toxin comes from a bacteria, and this acts at the neuromuscular junction. This is Botox as we know it. Botox is typically used in cosmetic procedures to prevent wrinkles, and it's usually used in anti-aging regimens. Botox is neither an agonist or antagonist because it acts presynaptically. So what does that mean exactly? Well, in image A, this is showing us how acetylcholine is typically released in the neuromuscular junction. So let's say we have a presynaptic neuron and a postsynaptic neuron. We know that acetylcholine is carried in synaptic vesicles. Synaptic vesicles are going to be carried down to the active zone, and usually snare proteins are playing an important role during this procedure. I do want to note that we know that calcium performs exocytosis when acetylcholine is removed from these synaptic vesicles. The vesicles fuse with the membrane here due to snare proteins. And we have talked about snare proteins in the past and how they play an important role in getting the synaptic vesicles full of these neurotransmitters down to the active zone at the synaptic cleft. This is what a cell is normally doing. However, when you're exposed to botulinum toxin, Botox interferes with snare proteins. The snare proteins that normally help the synaptic vesicles fuse with the presynaptic cell membrane in this terminal area no longer function properly. So essentially, the neurotransmitter acetylcholine can't be released, and muscle fiber becomes paralyzed due to this prevention in synaptic transmission. Thus, there's no activity between the neuron and muscle, and thus no wrinkles. So since muscle contractions can cause wrinkles, you want to inactivate those muscles to smooth the skin and look younger. And that's why some people choose to get these Botox injections. Botulism is caused by several kinds of botulinum neurotoxins that are produced by the growth of a bacteria called C. botulinum. And this is found in improperly canned foods. Essentially, the name for this bacteria comes from the Latin word for sausage, because of its early association of the disease that came from poorly preserved meat products. So botulinum toxins are very potent blockers at the neuromuscular junction, and it's estimated that as few as 10 molecules of this toxin are enough to inhibit a cholinergic synapse. So thus, this can be a very powerful neurotoxin. In some ways, it's been shown to hurt people through disease, through poorly preserved meat, and interestingly, we have now used this neurotoxin as a cosmetic procedure for wrinkles. So interestingly, most people don't realize that they're injecting a neurotoxin in their face when they're getting Botox. Another poison that works at the presynaptic terminal is black widow spider venom. Black widow spiders are actually commonly found in Southern California, so make sure you watch out for these guys. But essentially, they function similarly to Botox, but in a different way. When you're bit by a black widow spider, it allows a whole bunch of calcium to enter the neuromuscular junction. We know that calcium induces exocytosis. This causes the terminal to swell with all of those synaptic vesicles filled with neurotransmitters, and a huge influx of neurotransmitters are thus dumped into the synaptic cleft. The synapse becomes inactivated due to this overexcitation and influx of neurotransmitters. This is why you can die from a black widow spider bite. So if you ever think that you were bit by a black widow spider, make sure that you get to the hospital as quickly as possible. Next, we're going to talk about noradrenergic neurotransmitter receptors. Now, noradrenergic receptors are also called norepinephrine receptors. And this is because noradrenaline is the British term for norepinephrine. Norepinephrine binds to these receptors, and later on in this class, we're going to talk about how norepinephrine plays a role in the sympathetic nervous system response. But for today, we're just going to talk about how these receptors work. So there are two different types of noradrenergic receptors. First is the noradrenergic receptor beta, which induces excitation in the postsynaptic neuron, and the noradrenergic receptor alpha, which induces inhibition in the postsynaptic neuron. Now, norepinephrine binds to both of these different types of receptors we're going to be talking about. Interestingly, the noradrenergic receptor beta plays an important role in learning, memory, and cognition. It helps to strengthen synapses to promote long-term postsynaptic changes. And that's because it utilizes the second messenger pathway. So if you remember when we talked about G protein coupled receptors, I discussed there were two different mechanisms in which G protein coupled receptors typically work in the cell. They can either influence nearby neighboring ion channels, or they can induce a second messenger cascade. And this second messenger cascade typically leads to long-term postsynaptic changes that can be more permanent. For example, it can influence gene expression. So let's talk about how the noradrenergic receptor beta works. So essentially, the neurotransmitter norepinephrine can come and bind to the G protein coupled receptor. And we know that this G protein coupled receptor is made up of multiple subunits, the alpha, beta, and gamma subunits, and it's anchored to the phospholipid membrane with these lipid anchors. When norepinephrine binds to the receptor, it's going to induce a conformational change. The G protein can then activate the enzyme adenyl cyclase. Adenyl cyclase is an enzyme that plays an important role because it converts adenosine triphosphate, ATP, the cell's main energy source, into a second messenger known as cyclic adenosine monophosphate, or CAMP. Essentially, you can think of CAMP as a key second messenger because it's going to activate protein kinase. Remember that second messengers play a really important role in this long-term postsynaptic change. And because this whole process takes a longer amount of time compared to the process of a transmitter-gated or ligand-gated ion channel, it's going to affect the cell for these long-term postsynaptic changes. So once CAMP is created from ATP, CAMP activates another enzyme known as protein kinase. Protein kinase is very important because it can actually phosphorylate nearby ion channels. In this case, protein kinase phosphorylates the potassium ion channel and causes it to close its gates. When this gate is closed, the membrane potential will remain more positive or excitable because the cation has not left the cell, and thus it increases excitability of the postsynaptic neuron. I also wanted to provide you with another visual of how signal amplification of our G protein coupled receptor works. So when a transmitter binds to a G protein coupled receptor, the G protein is activated and it can stimulate the release of adenyl cyclase. Remember, this is an enzyme that converts adenosine triphosphate, the cell's main energy source, to CAMP. Remember that CAMP is important because it's going to activate protein kinase A, commonly referred to as pKa. And pKa has the ability to phosphorylate the potassium ion channels. This increases the amount of potassium that stays inside of the cell and thus plays an important role in neuron excitability. The second messenger CAMP can influence gene expression and help strengthen synapses. These are important for long-term memory. We'll talk more about CAMP in chapter 25 when we discuss the neural mechanisms of learning and memory. Next, I'm going to talk about the noradrenergic receptor alpha, which also allows norepinephrine to bind to it. So, this utilizes similar mechanisms to the noradrenergic beta receptor. However, it decreases the activity of the enzyme adenyl cyclase and thus makes the cell membrane less excitable. It prevents this cascade of actions, and potassium may still have the ability to leak out of the cell. We know that there are leaky potassium channels on the cell membrane, and thus this could lead to a more negative membrane potential. Because the membrane potential stays negative, it induces inhibition. So the key takeaway I want you guys to understand about this receptor is that when norepinephrine comes and binds to it, it induces inhibition in the postsynaptic neuron. And this is due to a decrease in activity of the enzyme adenyl cyclase. Make sure you check out both of these receptors that I just discussed. They're included in my handout sheet on Canvas. So next I'm going to talk about GABA neurotransmitter receptors. Now remember that GABA is an amino acid neurotransmitter and it's a small molecule neurotransmitter. GABA A is a ligand-gated ion channel, and it mediates almost all synaptic inhibition in the central nervous system, while glycine is another amino acid neurotransmitter that mediates the rest. The need to control inhibition may explain why GABA receptor has, in addition to its GABA binding site, several other sites where chemicals can dramatically modulate its function. And we're going to talk about certain drugs that can do so. But before we do, let's talk about how the actual receptor functions. Again, GABA A mediates most synaptic inhibition, which allows the neurons to rest. So remember that synaptic inhibition must be regulated very highly because if we have too much, it can cause a loss of consciousness or even a coma. However, if we have too little synaptic inhibition and too much excitation in the neurons, it could lead to a seizure. So we know that several drugs can affect GABA receptors, and I'll talk about that after we talk about its function. So here is from the handout that I drew for you guys, and we know that the GABA A receptor is a ligand-gated receptor. It's ionotropic, meaning that when the neurotransmitter GABA binds to the receptor, it can fluence ions because it is an ion channel itself. When GABA binds to the GABA A receptor, it's going to allow extracellular chloride to rush inside of the cell. And this is going to induce a negative charge because chloride is an anion. So thus, chloride makes the inside of the neuron more negative and generates inhibitory postsynaptic potentials. So thus, the binding of GABA to the GABA A receptor induces an inhibitory response in the postsynaptic neuron. This reduces activity in the cell. So as I previously mentioned, the GABA A receptor mediates most synaptic inhibition in the central nervous system. However, several drugs can actually bind to this receptor. Benzodiazepines such as Valium or Xanax and barbiturates like pentobarbital are selected for the GABA A receptor. Now, benzodiazepines actually increase the frequency of the GABA A channel openings, whereas barbiturates increase the duration of channel openings. Barbiturates like pentobarbital are used as a sedative. Sometimes they're prescribed for patients with epilepsy. However, high doses are deadly. Recreational use of any of these drugs and improper doses can be fatal. And this is because they increase more inhibitory chloride current and stronger inhibitory postsynaptic potentials. The behavioral consequences of this are enhanced inhibition. And we know that too much inhibition could lead to a loss of consciousness or even potentially a coma. So this is how many of these different types of drugs can actually affect the GABA A receptor and increase chloride movement into the cytoplasm of the postsynaptic neuron. Now, the GABA B receptor is also a GABA receptor, but it's a G protein coupled receptor. So, thus, it's a metabotropic pathway. So, when GABA binds to the G protein coupled receptor, it induces the shortcut pathway. So, rather than utilizing a signaling cascade that's going to result in a second messenger, it can actually just influence the opening of ion channels of these neighboring ion channels and allow this ion channel to open its gates. When the neighboring ion channel opens its gates, it allows potassium to leave the cell. And we know that potassium is a positive cation, so when it leaves the cell, it's going to induce a more negative membrane potential. Thus, this induces inhibition in the postsynaptic neuron. Now I'm going to talk about two key types of glutamate neurotransmitter receptors. The two key types include AMPA, which is a ligand-gated receptor, and the NMDA receptor, which is special because it's both ligand and voltage-gated. So both AMPA and NMDA receptors both sit and work simultaneously together. You can think of them as partners in crime. So they're both glutamate receptors, and they're both activated in the presence of glutamate. They both induce excitation in postsynaptic neurons. Both AMPA and NMDA receptors mediate the bulk of our fast excitatory synaptic transmission in the brain. So it's important to remember that both AMPA and NMDA induce excitatory postsynaptic potentials in neurons. Based on our previous knowledge from this course, we know that an action potential that ascends down the presynaptic neuron will end in the axon terminal, and this is going to generate exocytosis, when calcium is going to induce the release of the neurotransmitter glutamate from synaptic vesicles. When glutamate enters the synaptic cleft, it could then bind to the receptors on the postsynaptic neuron. And the receptors on the postsynaptic neuron that glutamate can bind to include AMPA and NMDA. The AMPA receptors are shown in blue, and the NMDA receptors are shown in pink. Glutamate binds to the AMPA receptor and then the NMDA receptors in the postsynaptic cell. When it binds the AMPA receptors, it induces the release of extracellular sodium that can now enter the neuron. And it's also going to allow sodium and calcium to enter through the NMDA channels. Ultimately, this increase in our positive cations is going to induce depolarization in the postsynaptic cells and thus generate excitatory postsynaptic potentials. Now remember, excitatory postsynaptic potentials are different from action potentials. They're a type of graded potential, and they can vary in size and frequency. However, we do know that multiple excitatory postsynaptic potentials can work simultaneously, and through this form of summation, they can potentially generate an action potential if the stimulus is strong enough. Here's an example of the AMPA receptor. You can also find this in the handout I posted on Canvas. Remember that AMPA is only ligand gated, which means that it's going to open when glutamate binds to its receptor. And this is going to allow extracellular sodium to enter the cell and depolarize it, thus generating an excitatory postsynaptic potential. Now, AMPA depolarization is really important because when AMPA depolarizes the neuron, it's then going to allow the NMDA receptor to activate. Even if glutamate's present, the NMDA receptor can't open until it reaches the correct membrane potential. So let's take a look at that in the next slide. We know that NMDA-gated channels can cause excitation in the postsynaptic neuron, and this is through the admission of extracellular sodium. But they differ from AMPA receptors in two very different ways. Make sure you know this for the exam. First, NMDA-gated channels are permeable to calcium, whereas AMPA receptors are not. Inward ionic current through NMDA-gated channels is voltage dependent, meaning that the cell needs to reach a positive charge in order for glutamate to properly bind and activate NMDA channels. We already know that calcium plays an important role in the presynaptic neuron because it induces exocytosis, the presynaptic release of neurotransmitters. Postsynaptically, calcium can also activate other enzymes that help to regulate the opening of different channels and ultimately affect gene expression. So, the activation of NMDA receptors can, in principle, cause widespread and lasting changes in the postsynaptic cell. And calcium entry through these NMDA-gated channels can cause the changes that lead to long-term memory. Now, we're going to talk much more about these changes when we reach our final chapters in this class, but I just wanted to put this on your radar. Now, as I mentioned previously, the NMDA receptor is both ligand and voltage gated. So, what does this mean exactly? Even if glutamate comes and binds to the NMDA receptor, it may not be able to open. And that's because it's probably at rest. When the cell is at rest at around -65 or -70 millivolts, there's a magnesium pore that blocks the channel. This magnesium block prevents other ions from passing freely through the NMDA channel. So thus, when the cell is at rest, even if glutamate binds to this receptor, there's no action that will take place. That's why the NMDA receptor is both ligand and voltage gated. So, if the cell becomes more positively charged, this means that the NMDA receptor can be activated. Magnesium can then leave and unblock, allowing positive cations to come into the ion channel. So, this can only occur when the membrane is becoming depolarized. An inward ionic current through the NMDA channel is voltage dependent in addition to being transmitter gated. Thus, this glutamate needs to bind in addition to depolarization in order for the channel to open and become activated. So both glutamate and depolarization must coincide before the channel will pass current. When this finally occurs, calcium and sodium can rush into the cytoplasm of the postsynaptic neuron. The brain uses a really large amount of our body's oxygen. It utilizes about 20%. And so, interestingly, in a disease state, oxygen deficiency can initiate a vicious cycle of excess glutamate release. So, if we don't have enough access to oxygen, we're going to induce an increase in glutamate release, and we can't generate enough ATP without oxygen. Remember that ATP is the main energy source in the cell. And in order to keep ion pumps working hard, like the sodium-potassium pump that's usually helping maintain homeostasis of ions in and out of the cell, membranes need to depolarize and calcium leaks into cells. Due to this depolarization, the entry of calcium triggers the synaptic release of even more glutamate. And glutamate further depolarizes neurons, raises intracellular calcium levels, and more glutamate is released. Now, this is the case of an ischemic brain injury. Now, as you can see from this paper by Lee and colleagues that was published in 2011, they show all of the different mechanisms in which excess calcium release can lead to neuron death. I just want to focus on this one right here. As you can see, an increase in glutamate release can ultimately activate glutamate receptors like AMPA and NMDA receptors, which trigger the synaptic release of glutamate through the entry of calcium. Glutamate further depolarizes neurons and overexcites them, and this activity can lead to cell death. So thus, oxygen and glutamate play an important role in ischemic brain injury. Now, the NMDA subtype of the glutamate-gated channel is really critical for excitotoxicity because it's the main route for calcium entry. Remember, the AMPA receptor does not allow calcium to come into the postsynaptic neuron. Neuron damage or death occurs because of stimulation by calcium of intracellular enzymes, and this degrades proteins, lipids, and nucleic acids. We know how detrimental this could be, especially since nucleic acids are the code for our genetic material, DNA. This occurs in certain neurodegenerative diseases. This is the case in ALS, which is a progressive disease that destroys nerve cells in the brain and spinal cord, which control the muscles throughout the body. When these nerve cells, also known as motor neurons, die, the brain can no longer send messages to muscle fibers that normally result in motor movement. And so this affects muscle movement. Alzheimer's disease is another example of how the NMDA receptor can play a role in the degradation of proteins, lipids, and nucleic acids. In Alzheimer's disease, neurons slowly die over time. For example, one of the main neurons that dies in Alzheimer's disease is cholinergic neurons. Cholinergic neurons typically release acetylcholine, commonly found in the basal forebrain, and these neurons can extend to the hippocampus. We know that the hippocampus is responsible for the neural mechanisms of learning and memory, and that's why Alzheimer's disease can affect memory. Neurodegeneration has been linked to decreased synaptic plasticity. So what exactly does this mean? So we know that the NMDA and AMPA receptors play a crucial role in synaptic plasticity. As the main glutamate receptors in the brain, they're responsible for the consolidation of our working memory into long-term memories. We know that synaptic plasticity is the ability for our brain to create and wire new synapses via chemical synaptic transmission. So thus, these glutamate receptors are playing an important role in this process in which we consolidate our working memories to long-term memories. Interestingly, NMDA antagonists have been shown to interrupt learning and memory processes in rodents. We know that antagonists block the NMDA receptor. So why would you want to block the NMDA receptor? Interestingly, NMDA antagonists have actually been used as treatment for trauma or drug addiction. For example, some studies in clinical settings have demonstrated that NMDA antagonists can reduce traumatic memories. This could be really helpful for people that are suffering from side effects and symptoms from PTSD. For example, studies have shown that both veterans and women in civilized areas that have PTSD do respond well to NMDA antagonist treatment. In these settings, they typically reactivate a traumatic memory by having the research subject talk about the memory, and they're administered an NMDA antagonist during this time. A common NMDA antagonist that's used is called memantine, and memantine could be useful for treatment of PTSD and drug addiction. However, there's still much research that needs to be conducted. When I was a student at Vanguard, I participated in a research lab with Dr. Michael Hanna, and in his lab, we looked at the effects of the NMDA antagonist memantine as a potential treatment for drug addiction. Now, interestingly, the NMDA receptor is utilized in the formation of long-term memories, and drug-associated memories can develop through the NMDA receptor. For example, a certain context, such as the place where a person did drugs, or the drug paraphernalia itself, can induce drug cravings because the contextual cues provided through this room or objects can actually increase NMDA activation and increase the drug-associated memory. Thus, we wanted to understand how the NMDA antagonist could be utilized as potential treatment for methamphetamine addiction. We use this paradigm below called the condition place preference paradigm. We utilized rats in this experiment, and rats were administered chronic methamphetamine to form an addiction. After a few weeks of chronic methamphetamine administration in the white chamber, we did a preference test and found that animals wanted to spend more time in the white box compared to the black box because they associated this white box with drug administration. Thus, we utilized this place preference paradigm to see if reactivating drug memories with an administration of an NMDA antagonist known as memantine to understand how that could potentially reduce those drug cravings or drug-associated memories. We knew that memantine inhibited the actions of the NMDA receptor and prevented glutamate from binding to the postsynaptic neurons. Rats injected with memantine immediately after reactivation of drug-associated memories showed a gradual decrease in preference to the drug-paired compartment. So in the example I previously talked about, that would be that white compartment where methamphetamine was typically administered. So interestingly, we conducted this preference test and we allowed the animals to freely roam in both chambers. Remember, there was a tunnel in between the white and black boxes. We did an initial preference test, and as you can see, the bars in navy represent animals that were treated with the NMDA antagonist memantine, whereas the gray bars represent the animals that were treated with just a saline solution as our control group. We found after a few days of memantine administration on test day 3, animals that were administered this NMDA antagonist had reduced preference for spending time in the white box that was previously associated with drug memory. Thus, NMDA antagonists like memantine could potentially help reduce drug cravings by reducing those drug-associated memories. This was a really awesome research lab to be a part of, and I really feel strongly about using drugs like this for treatment of addiction. However, there needs to be a lot more research done on this, as most of these studies have only been done in rodents, but I know that there are human research groups that are trying to investigate all of these different therapies for drug addiction.